Hello. We're now into chapter 18, amino acid oxidation and the urea cycle. And this is actually the last biomolecule to aid in energy production, and it's the last of our catabolic processes before we start to build things up. We've been tearing them down, but now we're going to be building them up after this point. And so let's just go ahead and get started. So oxidative degradation of amino acids is really interesting in many ways, and it's very different than what we've been talking about in the past so far. Because now we have nitrogens, and so these have an additional atom that we haven't encountered so far in biochemistry and in metabolic pathways. One thing that's really interesting is that carnivores. Sorry about that. That's Miss Myrtle playing with her Woody the Woodpecker doll. We can blame her grandpa for that one. He gave her that for Christmas. God bless him. Okay, but one interesting thing is that carnivores actually, right after they eat a meal, can get up to 90% of all their energy from oxidizing the proteins, the amino acids from that. Okay, and so there are actually three different ways to do for amino acid oxidation, and one is just normal protein turnover. Proteins die, okay, or they need to be disposed of because, for example, whenever proteins get where they're no longer useful or they start to misfold, one example of that is like the prion diseases or amyloid formation with respect to like Alzheimer's disease and serum amyloid A plaque formation and things like that, to where if the proteins were not turned over, were not degraded normally, then you die, essentially. Another way is that we have a protein-rich diet, okay? Especially those who are carnivores, because unlike fats and sugars, we have no way to do long-term broad-based storage of amino acids. And then finally, just as we alluded to in the last chapter, is either under starvation or uncontrolled diabetes, your body's going to want to break down anything and everything it can to get energy, and that includes your proteins to break down to the amino acids. But again, the majority, up to 90% of the energy right after a meal, so not during times of periods of fasting, in between meals, but right after a meal, can come from amino acid oxidation. So I think I know who would be really, really proud of this, and this would be his portion of biochemistry, meat. And so, not faith integration, but at least it's Parks and Rec integration. Good old Ron Swanson. Okay, so if we think about the amino acid, what are the main parts of an amino acid that can be scavenged or used? So let's consider this. We have, what makes up an amino acid? Okay, so you have the C alpha. You also have the carboxylic acid, or the carboxylate. I'm going to draw as the carboxylate. Of course, the hydrogen itself is of no import. Then we have the nitrogen, which in its ionized form would be protonated. And then we have the R group, which that can change, from amino acid to amino acid. But if we just think about the backbone, what parts of this are actually going to be of great use? Okay. Let's see. So, one way to think about this is if you look at the backbone of the amino acid, what parts of it look similar to the other biomolecules that we've used to get energy from? That would be this portion right through here. Okay, so the carbon-based portion, and if they have, carbon-based portions on the R group as well, those can be oxidized. And then the carboxylic acid itself is already fully oxidized, but they can, or is already oxidized, but they can be oxidized. And so it can be broken down in order to go to, if it's 3 and 4 carbons long, it can go through to other metabolic pathways. I don't want to give away the answer yet. Other metabolic pathways in order to be utilized for energy. However, we're stuck with this over here. The old pesky nitrogen. Nitrogen is not oxidized by us for energy. So we have to find a way to get rid of the nitrogen or to utilize the nitrogen for other purposes. All right. So first, the amino acid has to lose this amino group. And it's going to form our good friend alpha keto acid. So make sure you recognize what an alpha keto acid is. Okay, so just briefly, let's review what an alpha keto acid is. The alpha keto acid is going to be the acid portion is talking about a carboxylic acid. And once again, if it's a carboxylate, it would be an O-. And it's an alpha keto group. And then this is the R. You really need to recognize, what does this look like? What other alpha something acids have we had? And once again, this is the alpha group here. But what happens if instead of the alpha keto acid, I want you to recognize, and you have to be able to figure out, this alpha keto acid can also be, where's the eraser? An alpha amino acid. So you need to try to recognize which alpha keto acid looks like or is the companion to what alpha keto acid, and alpha amino acid, okay? Hopefully you'll get the hang of it. We've already been doing this to an extent with pyruvate and alanine, alpha ketoglutarate and glutamate, oxaloacetate or OAA with aspartate, okay? That's very important. All right. Now, once you lose the amino group, there are 3 possible fates. Once again, for the 3 to 4 carbons, you can oxidize it down to carbon dioxide and water. You get all the energy out of it. You can break it down to small carbon units that can go to various pathways, or to what pathway, in order to get to glucose for the brain. Or they can enter other ways and to get to the citric acid cycle, because ultimately you're wanting to get as much energy out of this as possible. So this is a good overview of what we're going to be talking about. So for example, either we're going to be breaking down endogenous proteins that are within the cells to form their individual amino acids, or you can have a dietary protein that we just imbibed from and have it broken down to amino acids. We're going to remove the amino group. If we need to, it could be used for biosynthesis of amino acids, nucleotides, and other important amines that we use in vivo. Or it's going to be put into what we're going to see as a common intermediate here called carbamoyl phosphate to enter into the urea cycle, ultimately for excretion. For the amino acids, we're going to break down to the carbon skeletons, to the alpha keto acids, enter into the citric acid cycle, to oxaloacetate for gluconeogenesis. All right. So here's a good picture of the way that this breaks down. Let's say one way that we have is the amino acids that we've just ingested, and we're going to talk about how they get broken down from the proteins. So we have some generic amino acid here. We've got to convert it to the analogous alpha keto acid, And we're going to find out the way that we do that is you take good old alpha ketoglutarate that we've seen before, alpha Kg, turn it into glutamate. Glutamate is such an important player here. Okay? Then the alpha keto acid can go on and be used for oxidation purposes, for the citric acid cycle, things like that. Another possibility we have is we're going to find out that alanine from the muscle, and you have to think about the reason why the muscles would have a lot of alanine. Alanine from the muscle can go through the bloodstream, come into the liver, the hepatocyte, and then once again, alpha ketoglutarate can come along, take the amino group to become glutamate, and the alanine is converted into its alpha keto group, which is pyruvate. Where can pyruvate go if we need energy? It can go into gluconeogenesis. All right. Now, at some point in time, what's going to happen is that you can take, and we'll talk about the role of glutamine. So glutamine can come from the muscle, from other tissues, and how what its role is within the blood, and how glutamine can be converted into glutamate by losing the terminal amino group of the amide. And then we have this NH4. And now the NH4, depending upon the species, is going to be excreted either as just ammonium, urea, or uric acid. And so I want you to understand this. For those who've already taken, zoology or some type of animal science, this should be old news for you. But certain animals are called monotelic animals, and so mainly fishes and some of the amphibia. And so they just excrete their nitrogenous waste in the form of ammonia, or ammonium, whenever it's in solution. Most of us, humans and terrestrial vertebrates, we are ureatelic, and so we excrete our nitrogen waste in the form of urea, and then we have birds and reptiles that are uricotelic, and so they don't urinate per se, they excrete it in the form of uric acid. Okay? And I want you to notice the carbons in the uric acid and in the urea are highly oxidized. So why do you think that that's important? That's because we utilize all the energy that we can out of that carbon. We want to fully oxidize it to where we get as many reducing equivalents out of it as possible to get all the energy that we can. So we're being very faithful stewards of the energy that's within our carbon-based compounds. And so I think that this should be a good stopping point for right now for this first part of Chapter 18.